Plant tissue culture broadly refers to the in vitro cultivation of plants, seeds, and various parts of plants, like it can be an embryo, tissue, single cell, or protoplasm. The cultivation process is invariably carried out in a nutrient culture medium and aseptic conditions. Plant cells have certain advantages over the animal cells in culture system. Unlike animal cells, highly mature and differentiated plant cells retain the ability of totipotency, that is, the ability to change the meristematic state and differentiate into a whole plant. So, totipotency is the ability of a single cell to divide and produce all, all of the differentiated cells in an organism. Spores and zygotes are the examples of totipotent cells. The culture of plant tissue in vitro involves the following steps. First one, formulation of culture medium. Second is selection of explant. Third is sterilization of the explant. Then fourth one is inoculation of explant. Then fifth is incubation. The sixth step is transfer of callus and regeneration. And the seventh step is hardening or acclimatization. And eighth is the transfer of the plantlets to the field. Coming to the first one, that is formulation of culture medium. Plant cells and tissues require a proper nutrient medium for their growth and development. The medium must contain the following components, that is, a carbon source, and usually it is a sucrose. Then macronutrients. Macronutrients like nitrogen, phosphorus, potassium, magnesium, calcium, and sulfur. Then micronutrients like iron, manganese, zinc, boron, copper, molybdenum, and chlorine. And they also require organic supplements like coconut milk, tomato juice, potato extract or yeast extract, and also some vitamins. Then the formulation also require hormones like IAA, NAA, 2,4-D, kinetin, and they also require a gelling agent, and it is usually agar. The pH should be between 5.5 to 5.8, suitable for cell growth, so the pH of the medium should be adjusted. Different media are used for plant tissue culture. They are often named after the discoverer who formulated the medium. For example, MS medium. This was formulated by Murashige and Shug in 1962. B5 medium formulated by Gamborg in 1968, and SH medium formulated by Schenk and Hildebrand in 1972. The LS medium, then the selection of explant. Explants are any excised part of the plant to be used in tissue culture, like axillary buds, leaf, stem segments, root tips, shoot tips, anther, ovary, and even endosperm. Always young and healthy parts are selected as explant. Then, after selection of the explant, the second step is sterilization of the explant. These explants are sterilized by using different types of disinfectants like sodium hypochlorite, 2% silver nitrate, 1% calcium hypochlorite, bromine water, mercury chloride, hydrogen peroxide, etc. and different types of antibiotics. Next is inoculation of this explant. That is, after sterilization, the explant is inoculated. The sterile explant is inoculated on the surface of solidified nutrient medium and aseptic condition. And for this, laminar airflow can be used. And the next step is incubation. And this is done at 25 degrees with a relative humidity of 50 to 60% with 16 hours of photo period. After defined period of incubation, an unorganized and undifferentiated mass of cells called callus is obtained from each explant. In some cases, root and shoot directly develop from the explant after incubation. Next step is transfer of the callus and regeneration. The callus obtained from incubation of the explant is transferred into another medium and re-incubated. Plantlet regenerates from callus with the induction of root and shoots directly from the callus. And the next process or the step is hardening or acclimatization. This is a gradual exposure of plantlets for acclimatization or adjustment to the environmental condition. That is from controlled tissue culture AC room to the outer environmental condition. The transfer of plantlets to the field. After hardening process, the plantlets are transferred to either the greenhouse or the field conditions outside. These plantlets will grow into independent and healthy plants. The importance of tissue culture. A large number of plants have identical features to the parents can be produced by this method. And the adult plants can be produced within a short period of time in this plant tissue culture technique. And another advantage is many plantlets can be produced without seeds. And even healthy and disease-free plants can be propagated by this technique. And apart from this, endangered plants can be preserved or conserved by this technique. Thank you.